Thank you everyone for attending this webinar. In this session, I will talk about the design and the development of a bispecific antibody. During this presentation, I will first give an overview on the therapeutic antibodies and focus on the opportunities and the challenges of the current monotherapy. Aiming to addressing medical needs, we will then discuss the major benefits of bispecific antibodies and review current platforms for that. I will also introduce GenScript proprietary SMAB bispecific antibody platforms, which minimizes the manufacturer concerns of the current bispecific antibody platforms, while enabling bivalent and multivalent therapeutics. For the last part, we will show a case study for SMAB bispecific antibodies, from rational design to preclinical development. Therapeutic antibody drugs have recently experienced explosive growth. The success of a cancer immunotherapy, such as PD1 and CTLA-4, created a hope for cancer cure, and it triggered a second revolution that the majority of the pharmaceutical companies are devoting major research force and investment in antibody therapeutics. Therapeutic antibody function through multiple mechanisms of actions to battle cancers, including direct inhibition or apoptosis of cancer cells, like anti-HER2 antibody, Herceptin for breast cancer, or through ADCC and CDC, like anti-CD20 antibodies, Rituxan for lymphoma, or through immune cell activation and recruitment, like anti-PD1 and anti-CTLA-4 antibodies for multiple cancer indications, and also anti-CD19 CAR T cell therapy for leukemia, or through cytotoxin deliveries like antibody drug conjugates. Compared with conventional chemotherapy, monotherapy, especially in immune oncology field, has significantly increased the survival rates. However, it still faces many unmet medical needs in which combination therapy and a bispecific antibody can offer particular advantage. As you can see in the BMS Checkmate 067 Phase 3 study in advanced melanoma, combination of anti-CTLA-4 Yervoy and anti-PD1 Opdivo showed much improved median progression-free survival compared with either one alone. However, this enhanced efficacy was associated with increased grade 3 to grade 4 adverse effects, from less than 30% to almost 60%. With this in mind, both bispecific antibodies and combination therapy can address many unmet medical needs. However, bispecific antibodies can offer additional advantages. First, bispecific antibody can provide superior potency due to novel mechanism of action. For example, the first generation bispecific antibodies are mostly T-cell engagers, in which the underlying principle is quite straightforward: burning T cells to target tumor cells. Other novel MOA includes modulation of receptor signaling and the simultaneous targeting of multiple co-inhibitory receptors or checkpoints, like bispecific antibodies against both HER2 and HER3, or against PD1 and CTLA-4, as well as the third approved bispecific antibody, Hemlibra, targeting factor 9 and factor 10 simultaneously for the treatment of hemophilia. Bispecific antibodies can also target multiple epitopes under the same target for synergistic effect. Secondly, bispecific antibody can offer enhanced safety profile due to less off-target binding and presumably lower dose, as we've seen safety concerns in combination therapies. Thirdly, developing bispecific antibodies is more cost-effective as it only involves one molecule and save half of the investment in comparison with combination therapy. All of these advantages of bispecific antibodies makes them a potential powerful treatment. To date, there are only a handful of approved bispecific antibodies in the market, but there are hundreds of them in clinical trials. This is a short list of in-clinical bispecific antibodies strategies by many world-leading biotech companies and biopharmas, with multiple MOAs, including T-cell or NK cell recruiters, 2 ligands inactivation, 2 factor dimerization, targeting 2 immune checkpoints, and to break the blood-brain barrier or induce more internalizations. In the next few slides, I will talk about various bispecific antibody platforms and focus on the GenScript SMAb platform. This is a schematic overview of the different strategies used to generate bispecific antibodies derived from the antigen binding sites of 2 different antibodies. Symmetric bispecific antibody are generated by the assembly of antibody with unmodified heavy chain constant regions, such as by the heterodimerization of heavy chains from 2 different antibodies, or homodimerization of heavy chains extended by an additional binding site, result in bivalent or tetravalent molecules. Using modified heavy chains to force heterodimerizations, such as knob in the hole strategy, result in asymmetric bispecific antibodies. Alternatively, 2 different antibody fragments, such as a single chain FV, can be fused together to a non-immunoglobulin protein, such as albumin. Furthermore, 2 antigen binding fragments can be directly fused together, resulting in a small bispecific molecules. Finally, bispecific antibody can also be generated by chemical conjugation of 2 different antibodies. To date, there are about 200 different bispecific formats, and the most common 20 formats have generated various candidates in clinics, or even approved by FDA and EMA. Despite the numerous bispecific formats and the benefits that bispecific antibodies have brought to patients, there are 2 major concerns for this type of antibodies. First, as most of the current formats are over-engineered, they may have immunogenicity concerns, as our immune system is very sensitive to unnatural stimuli. Secondly, as these bispecific antibodies often have unnatural formats, they are not development friendly or having manufacture concerns, including product instabilities, lower expression level, and the complex purification process. With these 2 major concerns in mind, we have developed the GenScript SMAb bispecific format, which is a single domain antibody fused to monoclonal antibodies. Our concept is very simple, that we will use all natural components to achieve bivalent or multivalent purpose. In this setup, SMAb has symmetric structure without FC engineering, which brings many advantages. It has the same single-step protein purification process as IgG, and since it has all natural antibody formats, the developability of the SMAb antibodies are comparable to conventional monoclonal IgG antibody, such as higher yield and ability for high concentration formulation. Moreover, since we use a single domain as one of the partner, SMAb can be very flexible with the ability to be multivalent. This is a direct comparison of the SMAb with other major bispecific formats, including Zymeworks asymmetrical bispecifics, Ablink's nanobody, Genmab duobody, and AbbVie-DVDIG. Zymeworks asymmetric format with FC engineering may have immunogenicity issues, which will be tested in several clinical trials. Ablink's nanobody format faces the issue of short half-life, and they are developing several strategies to overcome this issue. Genmab duobody involves post-production processing to generate the bispecific antibodies. AbbVie's version may introduce a steric hindrance due to rigid conjugation of two antigen binding sites. The SMAb format uses a symmetrical structure favoring production and stabilities while preserving long half-life and minimizing immunogenicity, but we still have to test this in clinicals. In SMAb format, we use a single domain as one of the partner because, first, the size of a single domain is relatively small, so it can bind to the epitopes normally hidden from conventional IgG, and also better tissue penetration abilities. Secondly, the single domain antibody has only one domain. It is easier to engineer to other formats. Comparing to conventional IgG, most single domains are more stable and can tolerate high temperature that enables the antibody to be delivered through multiple routes instead of IV. Moreover, the flexibility of a single domain makes it suitable to various therapeutics purposes, including bispecific or multispecific single domain, targeting multiple targets or multiple epitopes on the same molecule at the same time to increase therapeutic potentials. For single domain generation, Phage Display library approach is probably the most popular strategy. Here, I show a general workflow for single domain discovery using immunized llama or camel. After immunization, Phage library will be constructed using the PBMC from immunized animals. Phage biopanning and patent FASIBA high-throughput platform is used to screen for binders with good expression, affinity, and thermal stabilities for downstream further categorization. In addition to the traditional immunized library, GenScript also provides a ready-to-use single-domain naive library with the size of 10 to the 10 for candidate screening as fast as 6 to 10 weeks. For monoclonal antibody generation, there are several different approaches available at GenScript. Hybridoma technology is the most popular and well-developed one to generate antibody leads with good developability. However, it may take longer and has to be humanized from rodent hybridomas if not working with expensive transgenic animals. Phage display approach can screen out numerous hits within a relative shorter time frame, but may encounter poor developability issues. Single B-cell approach is able to generate antibody leads within a couple of days to weeks, but comes with higher cost and relies on capability of high throughput antibody expression and evaluation. It is a good idea to utilize at least two approaches in parallel to maximize the success rate of lead generation. In the next few slides, I will talk about antibody leads optimizations for bispecific antibody generation, including humanization, affinity maturation, developability assessment, and immunogenicity evaluation. Due to high immunogenicity of murine and chimeric antibodies, humanization is required for rodent leads. Antibody humanization is the process of replacing non-human antibody frameworks with human ones. Successful antibody humanization depends on maintaining the affinity after replacing residues. In general, there are two kinds of humanization strategies: CDR grafting and germline shuffling. Here I show a general workflow of antibody humanization at GenScript. Antibody sequence is analyzed to select the best human germline acceptors for murine CDRs. Post-translational modifications are analyzed to avoid PTM hotspots of selected germlines. After CDR grafting, homologous structure modeling can be performed to identify back mutation residues and use high-throughput screening to identify antibodies with good affinity, expression level, and thermal stabilities. Here is an example of a humanization of anti-cytokine single-domain antibody. We selected germline with higher sequence homology, and after homology structure modeling, we identified 16 framework residues that were different, highlighted in red, of which 5 residues were modeled to be potentially critical for the antigen binding, and the putative back mutations were performed. After this humanization process, we successfully obtained humanized single domain with comparable affinity and functions. Affinity maturation is another lead optimization process which aims to increase the affinity of the selected antibody leads. Usually, leads from hybridoma approach have reasonably good affinity with KD in nanomole to sub-nanomole range, and therefore antibody affinity maturation may not be necessary, especially with affinity guaranteed humanization process. However, in some cases, that the obtained leads may come with relatively low affinity, such as from phage library approach or transgenic animals, or the final application needs extremely high affinity. Affinity maturation is required. Here we show the flow chart of affinity maturation process. After the antibody sequence is obtained and confirmed with binding to antigen, we will perform the paratope mapping to identify the key residues within CDR for antibody-antigen interactions. An NNK single-point mutation library is built and screened to select top binders. Then, a combinatorial library is built and screened to select the best binders with the combination of mutations. After this process, the affinity will be increased at least 5 to 10-fold, and we have seen cases of increasing by 200-fold. For therapeutic leads selection of optimizations, antibody drug developability assessment is very critical. We mainly focus on the chemical stabilities, physical stabilities, and the solution properties. One key developability risk is post-translational modification, such as deamidation, isomerization, oxidation, free cysteine, N-link or O-link glycosylation. Various tools are utilized to assess developability of risk, including in silico structure modeling, size exclusion chromatograph, peptide mapping by SEMS, and affinity by SPR-based vehicle. For developability assessment, we can use these methods for biophysical characterizations, including SEC-HPLC, DSC to check the thermal stabilities, aggregation, freeze-thaw stabilities, biomatrix stabilities, expression level, and solubility properties. For PTM hotspots identifications like deamidation, isomerization, we can use peptide mapping followed by LCMS and check binding by SPR. In case there are developability risks identified by this method, Antibody engineering will be further implemented to remove or mitigate the risk. Immunogenicity of a therapeutic antibody is caused by various product-related and patient-related risk factors and can cause different therapeutic outcomes in terms of the drug efficacy and the patient safety. Various methods have been established for measuring and predicting immunogenicity, and FDA has guidelines for risk-based mitigation efforts for immunogenicity. There are several layers for immunogenicity risk assessment, including in silico prediction of T-cell epitope and the B-cell epitopes. Then multiple in vitro and ex vivo assays will be developed to evaluate the in silico prediction, including DCMS assay, PBMC assay, and whole blood assay. Lastly, non-human primates will be used to test the immunogenicity of therapeutic bispecific antibodies in vivo. Anti-idiotype antibodies are often developed to be used as a reference standard or positive control in immunogenicity assessment. In the next few slides, I will give you an example of a bispecific antibody, SMAB bispecific antibody, from rational design to preclinical development. We have selected PD1 and the CTLA-4 as the bispecific targets. Even though the detailed function mechanism of a PD1 and the CTLA-4 antibodies are not entirely clear, anti-CTLA-4 antibodies are believed to block the inhibitory signaling between antigen-presenting cells and the T cells during priming phase of T cell activation, while the anti-PD1 antibodies releases the negative regulation between effector T cell and the cancer cell, both of which can enhance T cell effector activation. We started with the sequence of a Keytruda and another anti-CTLA-4 single domain. By fusing the single domain to the IgG with linker peptide, we managed to get antibodies with 4 different formats. The single domain fused to the N-terminus of the heavy chain, the C-terminus of the heavy chain, or the N-terminus of the light chain, and the C-terminus of the light chain. By comparing the binding affinity, ligand receptor blocking capabilities, cell-based functionality, and certain developability indicators, the one with a single domain fused to the N-terminus of the heavy chain showed best outcome and was chosen to move forward. Here is the data summary and a quick comparison of in vitro properties between bispecific antibody and its parental antibody. This SMAB molecule successfully retained its activity throughout all 4 different aspects, including affinity measured by SPR, cell binding affinity measured by Phage EC50, ligand receptor blocking measured by Phage IC50, and functionality determined by cell-based signaling induction. In addition, we also compared certain developability indicators between SMAB and IgG, including the expression level in the transient transfection of a CHO cell, the purity after one-step protein A purification, the thermal stability is measured by DSC, the aggregation temperature, tissue cross-reactivity, non-specific binding, and freeze-thaw stability. These SMAB molecules demonstrated comparable developabilities as typical IgG monoclonal antibody. Besides the in vitro evaluation, we also tested this SMAB molecule in 2 separate in vivo models. In the A375 melanoma model, the candidates were tested side by side with the same molar dose of a single drug, combo, and IgG4 as a negative control. The data demonstrate that this SMAB candidates show better efficacy comparing to both combo and single drug. Similarly, in the MC38 model, both SMAb and combo show better response comparing with single drug. As I mentioned earlier during this talk, most of the bispecific antibodies in the current market is not development friendly due to its over-engineered structure. However, in our SMAb platforms, once we have a defined IgG and a single domain sequence, it will take us about 3 to 5 months for format screening, including both in vitro and in vivo studies. After that, we just need to follow exactly the same workflow as IgG for the preclinical CMC development. To summarize what we have covered today, bispecific antibody has demonstrated its superiority over mono and combination treatments. And the GenScript SMAb is a bispecific multivalent platform with open access. Its natural structure gives it better developability over other formats. Thank you all for your attention. Now we're ready for questions.